about B vitamins today. So my title is Back to the Future, and you'll grasp that a little bit better as we go along. But Back to the Future, B vitamins and their revitalized interest in the ocean. Um, Josephine just pretty much covered this whole slide that I had for you. So that was awesome. But I will say, at least this is where I normally sit. I'm not sitting there right now, but this is on campus here at Jordan Hall. And for a little bit of geo-referencing, this is literally where Raleigh is, NC State is, and the USA with the North Carolina. All right, so we'll go right through that and get to the outline here. So this is what we're going to talk about. I'm going to ask you all, what is a vitamin? All right, we're going to know that for sure by the end of the lecture today. But we're going to talk about, in particular, the big three B vitamins in the sea. You'll know about them. I'm going to cover a brief history of B vitamins because it's kind of fun and interesting. Then we're going to shift to covering auxotrophy and prototrophy, and then get into some bits of more recent research focused on B vitamins and kind of why people are so interested in them once again. All right, so like I mentioned before, any questions, interject, okay? No problem there. So first question here, what are vitamins? Does anybody know what a good definition is for a vitamin? I've given you three little pictures here that might inspire some ideas.
Vitamins are organic compounds that are needed in small quantities to sustain life.
Exactly right. You might have been using Google there, which is also also fine. That's totally fair game. Exactly right. So everybody get that? So kind of like trace metals, right? You only need small amounts of them. Vitamins are organic nutrients though, so they're not metals. So exactly right. They're kind of dynamite in small packages. You only need a little bit of them to satisfy the need for cells. So the big three B vitamins that are best studied, I'll argue, in marine microbiology are these three. So that's B1, B7, and B12. They have these names: thiamine, biotin, cobalamin. They can structurally vary quite a lot and vary quite a lot in size as well. But these have been known about for decades, centuries, and are also added in many common media to grow marine microbes. For example, F over tooth. If any of you grow algae, it's a really common one. There are other vitamins out there. We just know the most about these. Okay, so maybe you'll go on and look into riboflavin and other vitamins that actually do exist, but I just want to make sure that you're exposed to at least these three here today. So what is the functional significance or importance of vitamins? Well, I'll argue the primary function for most vitamins is that they're enzyme cofactors. So enzymes are going to work much more efficiently at carrying out reactions when they have, for example, thiamine as a cofactor to carry out reactions. All right, I imagine all of you are quite familiar with cofactors, so I won't rehash that bit of biochemistry there. But more specifically, there's a number of steps in central metabolism that vitamins are required cofactors for enzymes that carry out these steps. And these are color-coded here, but this includes steps in glycolysis, the Krebs or TCA cycle, nucleic acid synthesis, amino acid synthesis, etc. So a lot of core steps require vitamins basically as a cofactor catalyst to get those steps done enzymatically.
I have a question.
All right.
So if you go back to the picture with the vitamin and the enzyme, that one,
yep.
Yes. So my question is, is the vitamin, is it like really important for the enzyme's function, or can the enzyme function without the vitamin but just in a worse way?
Yeah, exactly. So we'll get into a little bit more on this, particularly with B12, but for the vitamins, so there's enzymes that have literally evolved to have a dependence on these vitamins. And so in most of those cases, I'm unaware of those reactions actually happening without the vitamins. Now, it might happen really, really slowly. We'd have to dig in deeper on the biochemical literature there. But as you'll see with B12, there are some enzymes that that cells have or have evolved to no longer use vitamins as a cofactor. So to get past that requirement, that's not a widespread thing, but that does happen. Does that make sense?
Yes, thank you.
Yeah, no problem. So a few other functional significance or importance of vitamins that we know of are that they can also function as riboswitch ligands. If you're unfamiliar with this, this is an element that's on mRNA that actually kind of acts as a switch that regulates whether or not that mRNA signal is translated into protein. So it's another control for the cell to say, stop making this protein because there's plenty of this vitamin around. I think someone's put something in the chat. I'll try to. All right. Should I wait till the end, Rachel, to? Okay.
Yeah.
Sorry, Sebastian. We'll get back to it. I'm not neglecting you.
Okay.
All right. So hopefully the riboswitch concept makes sense. The other one is that vitamins can function as antioxidants, and we hear a lot about this in terms of supplements for our own diet. But I find that some people maybe are challenged by understanding that. Do you all understand the idea of an antioxidant and how that sort of functions?
I don't.
So yes, yeah, Rachel. Eric, I saw Eric nod his head. A few people said they didn't. So basically, if you did an in vitro experiment and you had a vitamin, but then another organic compound that will react with a reactive oxygen species, say peroxide or hydrogen peroxide, and you combine all those together, the vitamin will prevent transformation of that other organic. So it'll kind of shield oxidation of this other compound. So that's how you know it's a useful, effective antioxidant. All right. Okay. So this is kind of getting to Eric's question already here. We know that there are some cells that do not actually require vitamins. So some have maybe smartly evolved to no longer require vitamin B12, for instance. They don't have that requirement upon them. And a classic example here are members of the SAR11 clade, a really abundant bacterioplankton clade in the surface ocean. So they've evolved to have methionine synthase, an enzyme that normally requires B12, but to have an isoform or a form that doesn't require it. So they've escaped the need to acquire B12 that way. Okay, so there are some organisms that don't require vitamins. Actually, well, certain vitamins, I should say. Okay. So with that, I want to shift to the brief history of vitamins for you. Okay. Really, the first indication that vitamins existed, or that we realized that they existed, was based on dietary issues with humans. So we knew that if you supplemented with certain factors, we would improve in health. And the classic example is scurvy. Many of you maybe are familiar with it, especially if you follow pirate history. We have a famous pirate, North Carolina, Blackbeard, who I should say infamous pirate. He wasn't exactly the nicest guy in the world, but many times you see drawings or caricatures of pirates with loose teeth and maybe gum disease. It was later discovered that if you supplement your diet with citrus, those symptoms would decrease. And even further along, people realized or determined that vitamin C was actually that factor that's present in citrus that would help avoid that scurvy, basically. Okay, so at this point, it was just some sort of factor that we knew in certain foods that would allow people to avoid diseases. Now, boring into this a little bit more, this fellow Christian Ekman specifically determined that there was a factor in unpolished rice that reduced instances of beriberi, this circulatory and neurological disorder. Okay, and he figured this out through a pretty amazing experiment that I'm certain you could never do again, but this was done in Javanese prisons. So he tracked the diet of prisoners and also the prevalence of beriberi, and found that amongst the prisoners that ate unpolished rice, so this had components of the rice husk, a little less cleaned rice, basically, that they did have a lower incidence of beriberi, meaning that there's something in that unpolished rice that they were consuming, a factor that was allowing them to avoid this disease, basically. And from there, this researcher, Casimir Funk, who has probably the most amazing doctor name ever, Dr. Funk, I think, systematically figured out what fraction, either the lipid or water fraction, contained this this active ingredient, or this factor from the unpolished rice that was helping alleviate this disease. And so he named, so he did this chemical fractionation. He just did a lipid or a water hydrophobic hydrophilic extraction, named the lipid fraction A and named the water fraction B, and figured out, okay, this factor is in that water fraction, the B fraction. And so from there, he actually, from that separation, that was what led us to this naming scheme that we have today. So it turned out that factor in the water fraction was actually vitamin B1. And that's what he ended up determining, called it thiamine. And we've stuck with this naming convention. So the vitamins that are water soluble are the B vitamins. Okay. He also coined the phrase vitamins, so it's just an amalgamation of vital and amine, but that is also kind of stuck from his work. So just a very brief history there of early vitamin work. So I'll transition there to marine related B vitamin work. Okay. And specifically, we got to talk about prototrophy and auxotrophy. All right, make sure that everybody understands this. So prototrophs are the organisms that can make a vitamin from scratch, de novo. All right. Auxotrophs are those that need a supply of the vitamin to survive. They can't make it on their own, but they still have a dependency through those enzymes, essentially, to acquire the vitamin. We're over here in this auxotrophic side. Basically, all upper trophic level organisms are also there. We need a supply of vitamins. We can't make it ourselves. And what I think is kind of amazing and also important to mention is we as marine microbiologists knew about this this dichotomy that there's prototrophs, autotrophs decades ago. So even back into the 1940s, we knew that there's some cells out there in the ocean that are prototrophic and auxotrophic. How are people doing? That makes sense? So they used both bacterial cultures and algal cultures to figure this out. So through cultivation methods.
Could I just ask something quick?
Sure.
Are you a prototroph for a specific vitamin or prototroph for all vitamins? Like,
right. We'll get to that too, but usually, well, we now know that it's often that it's not uniform. You can be a prototroph for one vitamin and an auxotroph for another.
all right.
Generally, for us, we're auxotrophic for all of them. We have to eat foods basically to meet our demands. Does that make sense?
Yeah, sure. Thanks.
I have a little follow-up question, kind of.
Sure.
Prototroph, are that used for like prokaryotes, or can eukaryotes also be prototroph?
Good question. No, it's not just unique to prokaryotes. Yeah, certainly you can have eukaryotes and prokaryotes that fall under auxotroph and prototroph. Exactly right. Yeah. All right. So also in this early work, people were figuring out how to use these cultures to quantify B vitamins in seawater. So what they would do is use bioassays. So they would have an organism that was an auxotroph for a B vitamin, knowing that it needed to grow on supplied B vitamin to live. They could link the amount of growth or yield, so cells that were produced, to the amount of B vitamin that was actually in an unknown sample. So you have an unknown sample. As long as you know that the growth rate per vitamin present or yield per vitamin present, you can back calculate how much vitamin was in your initial sample. And a classic example here is this work by Carlucci and Cibernagel, where they're using this lovely green algae, this Pavlova lutheri, and basically running these experiments to look at yield, or they also use C14 incorporation, so kind of an indicator of phytoplankton growth there to estimate how much B vitamin is in seawater. And from those early studies, they were finding that there's picomolar concentrations of B vitamins in seawater. So pretty small. Do you all know what the concentrations of DOC are in, say, the upper ocean?
No idea.
No idea. All right, so it's micromolar. So we're talking 6 orders of magnitude difference in the total DOC, total organic carbon out there, and vitamins. So a huge difference. Again, these are dynamite in small packages. Little amounts go a long way. So there's only low concentrations present out there, and there's also very low requirements per cell, sometimes in the range of 100 molecules needed per cell. So this was determined all using early, in these early studies using isolates, okay, these bioassays. Now, there's a lot of other things that were discovered early on too, and we don't have time to go through it all, but a very quick crash course, people also were figuring out that cells readily provide B vitamins, when they're growing, to other cells. There's light and temperature degradation that happens of vitamins. We know that they figured out that pieces of the vitamins could actually suffice to sustain cells, not just the intact vitamin. And then also there was a few studies that were suggesting B vitamins could limit primary productivity, so carbon fixation in some parts of the ocean, particularly these HNLC regions. And I don't know if you all have talked about that before, but that stands for high nutrient and lower than expected chlorophyll levels. Okay, so a lot was done early on here. And what I want to get to is the fact that we're revisiting this now. And hence the name Back to the Future and Michael J. Fox here in the picture. But a lot of this has been sparked by our increased genomic data and cultivation efforts. So we've now cultivated a lot of organisms that we couldn't cultivate before. We now have a lot of genomic data that was definitely not accessible in the mid 1900s. And the other factor is that there's been novel chemical quantification methods developed. Okay, so these two factors have really pushed, sparked, revitalized interest in B vitamins. Okay, so I want to dive into these with you for the rest of the talk here. So on the increased genomic data and cultivation side, I think this is probably pretty intuitive, but there's many organisms now that we've cultivated or sequenced that were totally unknown back in the mid 1900s or late 1900s. And this includes both algae and bacteria. And one recent effort basically summarized auxotrophy and prototrophy based on isolate genomes. So kind of updating our knowledge on auxotrophy and prototrophy. Okay. And I thought this would be a good opportunity to kind of give you the basic paradigms on algal and bacterial auxotrophy and prototrophy. Because I just described before what it was. I didn't say kind of what's the common pattern that we see in the ocean. And so the summary here is that you can look to genomes and see whether or not certain genes are present, absent, and now predict whether or not they are auxotrophic or prototrophic. Does that make sense to everybody? So you sequence the genomes and you do what's called comparative genomics. Also, we know the components to the pathways because we have really good reference organisms like E. coli, where people have done knockouts and know that functionality is gone if you lose this gene, and we know what it does. So now you can compare your genome to the E. coli genome or annotations that are to proteins or genes there to predict whether or not it's an auxotroph or prototroph. People all good with that? All right, so what I'm showing here are algal genomes, so similar to the E. coli analogy, but the summary here, I don't expect you to look through all these tables, is that there are some algae that are B1 auxotrophic. All of these sequence genomes suggest that they're B7 prototrophic, so they produce B7, and none are B12 prototrophic, so they can't make B12 from scratch. But going back to Eric's good question there, way back in the beginning, there are some that are independent of B12. They have enzymes, they have the methionine synthase that doesn't require B12, so they've bypassed a B12 need. Okay, there's a lot of B's there. And people are doing okay still, not freaked out. All right, so I'll be able to share these slides. You can go back and reference this, but that's the algal summary here. So they also look to bacteria, and I'm saying small b here because we'll include archaea in this. Most of those bacteria are B1 and B7 prototrophs, so they're producers of B1 and B7. There are some auxotrophs of B1, B7, and B12 also scattered in there too. So they're not all just producers. Archaea appear to be commonly, specifically I should say, Thalarchaeota are B vitamin prototrophs, so producers of the vitamins. Okay, so in many instances, bacteria are key producers of vitamins, but we have evidence that there are auxotrophs out there as well. They're not just producers. And the last thing that I have here, this last bullet here, I should say, is that this is isolates. So this is, are you all familiar with the Great Plate anomaly? So, so these are what we can, largely what we can cultivate. So as it turns out, if we turn to using techniques like metagenomics, metatranscriptomics, we see that actually in natural communities, it's more complicated and can be different. So the distribution of auxotrophy and prototrophy is different from the patterns amongst cultivated strains. Does that make sense? Because you're looking at what's actually out there and not depending solely on what's been isolated and cultivated. Okay. There's a lot here, but I'll just go through the bullets with you basically to summarize. And they're using metatranscriptomics to do this. So they're looking at mRNA signals. And the conclusion here is that there's mosaic patterns. So there's a lot of phylogenetic groups that actually have mixtures of being B1 auxotrophic, let's say, and B12 prototrophic. It's not as clear cut as necessarily what the isolate information told us. So that means that there's probably a lot of interdependence, a lot of networks where there's cells that are sharing one vitamin, but then they're getting another vitamin in exchange, because there's this kind of heterogeneity of the types of auxotrophy and prototrophy. Okay, are people still okay? Not too dazed by all this? All right, there are some emerging trends here, and I won't dive into this, but too deeply. But one example is that rhodobacteria, for example, seem to be significant B12 producers, maybe more so than we originally realized. All right, so this, I wanted to bring this up. So we, as a postdoc, we actually did something similar, looking at metagenomic data from a Baltic Sea station. So this is the LMO station. It's off of Kalmar. And the short and sweet of this basically is that we focused on vitamin B1, and at the LMO station, and also it turned out a global surface ocean, most of the bacteria there are actually B1 auxotrophs. Now, I've mentioned a lot of things, but before with the isolate surveys, it looked like most were actually B1 prototrophs. So this is really kind of shifting our original thinking that actually a lot of these vitamins are a lot of these bacteria are actually cheaters for B1. They're auxotrophs. They're getting it from somebody else out there actually. And on top of this, I didn't want to show too much, but we also could see that there's a lot of different B1 related genotypes. So not only are they auxotrophs, but there's different types of auxotrophs and ways in which they take up vitamins through transporters or scavenge pieces. And we're yet to figure out the advantages of those lifestyles, but it's clear that there's a variety of them out there. It's not just as simple as they're all just plain Jane B1 auxotrophs. Okay. All right, so taking a breather here for a second. So there's a lot of exciting research that's come out focused on, well, that is showing the importance of vitamin precursors and analogs. So not just the initial vitamin that was discovered half a century ago. Okay, so that there's actually these variants on them. So they chemically are slightly different, or there's pieces of the vitamin that are quite important to sustaining environmentally abundant or ecologically relevant populations. Okay, So there's not enough time to go through all of this. I just want to give you a couple of examples. Okay, there's several papers that I mentioned here at the bottom that you can look up later. But one story I'll talk with you about is about Pelagibacter ubiquae. So this is a representative isolate of SAR11 bacteria. It's really difficult to cultivate. So a number of researchers worked really hard to make that happen. They sequenced its genome. And when they looked at this genome, they could see that there's a number of these pieces of the B1 biosynthesis or acquisition pathways that it didn't have. So that's these X's here. And it's a little tough to interpret, I would say, in the short term, but basically what that was signaling to them is that these Pelagibacter ubiquae cells cannot take up external B1 straight away because it didn't have this transporter. And they thought that likely it's using this precursor because it does have this transporter, Thi-V. So with the ability, with this organism in culture, they could actually do growth experiments and supplement, improve or disprove this hypothesis that it required this precursor HMP to survive. And that's indeed what they were finding, is when they added HMP, it would actually grow under B1 vitamin limiting conditions. So this is a unique find because actually this organism only survives by using a precursor compound, not the intact vitamin. Okay. People follow that all right? Yeah. Okay. Another example that I wanted to briefly mention here on the cultivation genomic side is with B12. So this Helliwell study was actually using a number of cultivated strains, cyanobacteria and eukaryotic algae, several which that had sequenced genomes, and they were actually finding that the cyanobacteria that they use in their experiment actually produce only this pseudocobalamin. So it's a slight variant of B12. Okay, it's one that has a different lower ligand to the B12 molecule. But the important thing here is that the eukaryotic algae that they use in their experiments would not grow on this form, at least not particularly well. And instead, it grew on cobalamin, this form here that has a different lower ligand. Okay, so these are just slight chemical modifications to the molecule. And so they basically show the importance of these different molecules, and that for eukaryotic algae, it's going to be very important that they obtain this form of B12. Okay, because they can't grow on the form that's provided or produced by cyanobacteria. And if they are, they're going to need some remodeling of that molecule. Okay, it's going to need to be changed into this cobalamin form before they can actually use it. The last bit with this study is that they actually found there's some strains of eukaryotic algae that could just use that lower ligand piece, and they made the rest of the B12 molecule and met their requirements that way. So this is another example of where we're disentangling all these details in terms of the forms of vitamins and pieces of them that are actually important to environmentally abundant or significant populations. All right. Everybody still with it? I see there's 17 comments here, so there's going to be a lot of questions here at the end. So I better speed up. So last bit I promised to discuss was the chemical measurement side. Okay. Specifically, this uses LCMS. This stands for liquid chromatography mass spectrometry. All right, so this is just chemical measurement. Instead of using bioassays, chemically measuring vitamin in samples. And this can be dissolved seawater, or this could be biomass that you collected. With dissolved seawater, you run the seawater through a resin and it captures organic compounds there, and you extract it and then use this mass spectrometry to measure how much vitamin, your exact molecules, are in that extract. Okay, so this enabled us to, or enabled people to generate profile data of dissolved B vitamins, which is shown here in the bottom. And specifically, Suffrage and colleagues have really made a lot of headway on this, and have obtained a lot of oceanographic profile data for B vitamins, both in the dissolved and particulate phase. And the utility of this is that you can start to improve predictions of biology out in the ocean. So in this case, they're showing that you can actually better predict bacterial abundance if you include B vitamin measurements along with inorganic measurements. And also, it's really improved estimates of of cellular vitamin content. So I wanted to ask or pose this question: why do you think having improved quota numbers is good? You know, why is that useful to us?
Right, maybe you can explain what you mean by quota here.
Sorry, so yeah, that would be vitamin per cell. So how much vitamin is in a cell actually in the ocean?
Mhm.
Why would it be important to know that in kind of an ecosystem sense?
I was thinking like it is important to know because it's good to know which vitamins that are really important for the community and which one that can be prevent growth in some species or in the primary production.
Yeah, so that's right, to know which ones are really available. And actually my next slide was on that. That's for sure is true, but also think back to the auxotrophy prototrophy side. All the upper levels of the food web basically need to consume cells that are lower in the food web, right? So most of their vitamin supply is coming through cells. So if we're going to budget better how much support is coming up the food web through cells, we want to have good numbers for those prey, basically. Does that make sense? So a lot of grazers and produce out there are like us. They need to eat food to meet their vitamin demands. Okay.
So
All right.
sorry.
Yeah, Fabian.
Just to clarify, so it would be useful for like food web insights into food web. Is that right? Or
Exactly. So budgeting vitamins within the food web and possibly predicting whether or not there's food web impacts. That's
Okay.
certainly important. Yeah. Good question. So last bit here with the LCMS side is just, I want to also mention this study from Heal et al. That looked at this B12, looked at the different variants of B12 that I just mentioned, which might have been difficult to grasp, but there's these variants again that have different ligands at certain parts on the B12 molecule. And what they are showing basically is that cyanobacteria, similar to the other study, they knew that cyanobacteria only produced this certain form, and other bacteria and archaea would produce another form of B12. But this had not been looked at directly in the ocean. And so they did this and were actually able to elucidate really interesting patterns where they could indeed find both of them in the particulate matter. So that was back to what Eric was mentioning, being able to actually know what's out there. And indeed, both of those forms of B12 were there. But in particular, they pinpointed that these common cyanobacteria are the source of this pseudocombalamin, and that heterotrophic bacteria and archaea are really key cobalamin producers. And this form, again, is the form that eukaryotic algae really like to grow on. Okay. So to wrap up here, I'll just say we've learned a lot in these recent studies, but there's a lot of ecological questions that remain unanswered. And one in particular here is whether or not these B vitamin concentrations ever limit microbial productivity or growth. You know, there might be just enough of them out there, precursors, pieces, all those complicated things that I've tried to cover with you today, that everything's fine. They're growing at close to their maximum rate. We don't know this exactly yet. There's better overt concern from studies related to animals, and the Baltic Sea is a good example. Some work in the Baltic Sea, excuse me, is a good example of this, where we know that there's fish and birds that are suffering from B1 deficiency. But so far, there has been some recent studies that are arguing, okay, in some instances, B vitamins may actually limit microbial productivity, be it bacterial production or primary production, but we need more studies to really thoroughly look at this, okay? So I've already gone over time here, and so maybe I should stop and just leave these take-homes for you to read on your own time. Is that probably best, Rachel, so we can get to all 17 chat questions? I'm